afternoon, everyone. Can you hear me? Good afternoon, everyone. My name is Nour, and I'm a first-year PhD student at the Kabashi team. And my talk, it's not. Oh, is it working? Can you hear me? I'm not sure what's. Hello. Yeah. This is. I should sound like this. Okay. I don't think. Is it okay? Good afternoon, everyone. My name is Nour, and I'm a first-year PhD student at the Kabashi team. In my talk today, I will be presenting our work on FUS-mutated ALS, demonstrating how it is involved in metabolic homeostasis and discussing clinically relevant therapeutic strategies using the zebrafish model. First, I will start by giving a brief introduction on the fused-in sarcoma gene mutation. As you might already know, it is the primary cause of juvenile ALS, where FUS-ALS patients display skeletal muscle atrophy and also FUS mislocalization, where FUS becomes impaired in the nuclei. So besides those neuromuscular junction features, data have shown in the past years that FUS is also involved in abnormalities in the mitochondria, which lead to impaired lipid metabolism. So back in 2020, in our team, we have characterized a FUS knockout zebrafish model, replicating the FUS ALS key pathological features. As you can see here from the survival curve, there was a decrease in the survival rate of the fish, which is also shown to have an increase in the larval mortality. So to further validate the FUS mutation expression in our model, we looked at the locomotor defects seen in the zebrafish. First, we analyzed the distance, velocity, and time the fish swam, and they were all reduced. In addition, we further confirmed this phenotype by studying the locomotive behavior at a later time point, and also confirms this phenotype. In addition, we saw some shorter neuron axons, which confirmed the presence of neuromuscular junction abnormalities. So in my presentation, I'll be focusing on two main aims. The first one is looking at the metabolic and transcriptomic alterations associated with FUS-ALS, by first looking at the morphological features that led to those alterations. And this work was mainly done by our previous PhD student, Dr. Mingaj. In addition, for the second aim, I will validate a pathway that can be targeted and identify new therapeutic strategies. First, we started our work by looking at the skeletal muscle structure. We evaluated the differences in skeletal muscle integrity, and this is by looking at the slow and the fast muscle fibers. And as you can see, we did immunohistochemistry staining, and we saw the differences in the myosin heavy and light chains, which were significant in between the different muscle fibers and amongst the genotypes. As well, we can see a misorganization in the alignment of the FUS minus, which confirms that there is a structural difference from the wild types, and a different alignment and orientation. The second structure we looked at was the mitochondria. We wanted to assess the morphological features of the muscle mitochondria. And this work was also done by immunohistochemistry. We used the TOM20 mitochondrial stain on whole embryos. And we were also able to see significant results on the mitochondrial morphology, where from the images we can see a pronounced disorganization of the mitochondrial network, in addition to some accumulations in the perinuclear region and some fragmentations in the FUS homozygous fish. So after we saw all these morphological differences in the skeletal muscle and the mitochondria, we wanted to learn more about the underlying molecular mechanisms causing those structural differences. So what we did is we performed RNA sequencing that was done at the genomics platform and at Institut Imagine. And the data has given us a great insight on what to look for next. So for the transcriptomic analysis has shown multiple downregulated and upregulated genes, as you can see here. And one of the striking results was the downregulated metabolic pathways, which we wanted to investigate further. Okay. So as I said, we did the metabolic analysis at the metabolic platform at Necker Hospital, and the metabolites and set enrichment analysis has identified significantly enriched KEGG pathways. And as you can see, they were mostly associated with amino acid carnitine metabolism and glycolysis. And from the figure, we can see one of the top enriched pathway is the transfer of the acetyl group into the mitochondria, which is linked to TCA cycle regulation and carnitine synthesis, which we decided to further investigate the metabolite levels. So what we did, we looked deeper into the metabolic pathways involved, and we analyzed major molecules such as the free fatty acids, carnitine and acetylcarnitine, and all of them showed a significant decrease. And those results suggest that there is an altered fatty acid transport into the mitochondria, leading to a potential incomplete or disrupted fatty acid beta oxidation. So then we went back to our transcriptomics data to try to find the link to the affected fatty acid beta oxidation we saw in our metabolomics. Interestingly, the PPAR alpha was one of the top pathways in the downregulated genes in the transcriptomics, and it governs various parts of the fatty acid metabolism. So it was our pathway of interest to check it further. But first, what is PPAR alpha? It is the peroxisome proliferator activated receptor alpha. It is responsible for lipid and glucose metabolism, and it's shown to be involved in multiple diseases, such as including neurodegenerative disorders. And finally, it's the major transcriptional regulator of fatty acids. So here we did the IPA analysis to look at the different genes involved in the PPAR alpha pathway and to see how they interact with our gene of interest, which is FUS. We saw multiple genes that were downregulated in the pathway, including an interaction with FUS. So this proves that the PPAR alpha pathway is a major pathway, and we decided to target it to potentially restore the function of the mutated FUS. Okay, so what I did at the lab, I decided to perform a rescue experiment to target the PPAR alpha pathway downregulation, and this was done in the FUS mutated zebrafish, where I injected the PPAR alpha plasmid in hopes to gain, the fish will gain back their normal phenotype. And this was done by injecting the plasmid at the 1-cell stage of the fish, and then we measured the locomotor activity. So this was done by the touch-evoked escape response, and we looked at the swimming trajectory of the fish. As you can see, the fish swam in a more straight, enhanced swimming pattern compared to non-injected. And also, they were able to swim longer distances, showing that the phenotype was rescued. Then we wanted to target this downregulation using a molecule that can help in restoring FUS gene function. So by looking at the previous literature, we identified a drug, which is called fenofibrate. It's a PPAR alpha agonist. And in the literature, it was shown to regulate PPAR. It is a member of the fibrate class of hypolipidemic drugs, and it reduced inflammation and promoted mitochondrial stability. Also, in the SOD1 mutant mouse model, it has shown to reduce inflammation and increase survival rate, among other positive effects. So that's why we decided to use it as a treatment for our model. So I tested the drug in our zebrafish FUS ALS model, and we were able to obtain promising results. We were able to see enhanced locomotion in the fish, and the swimming pattern was also significantly improved, as you can see here from the trajectory figures between non-treated and treated. And also the fish were able to swim longer distances, and this can prove that fenofibrate is a potential targeted therapeutic strategy. To sum up, it's shown that FUS has a great importance in muscle structure integrity and preserving mitochondrial function. In addition, the transcriptional and metabolic alterations we saw in our model confirm the crucial role of FUS in orchestrating those metabolic pathways. In addition, by targeting the downregulated PPAR alpha pathway, either via a plasmid or a drug, it was shown to improve the motor function and extend the lifespan of the fish. Hence, this can open avenues for new therapeutic strategies. Thanks to Arslav for this, and thank you for my team for all the help and special guide. Thank you, Dr. Mingash, for your special guidance.
Thank you very much. Viewing the progress time, we only have time for one question.
This is a very nice talk. We need to be very careful, caution, because the phenofibrate is not penetrating well the brain in human. And so it was very promising also for stroke, because you disrupt the blood-brain barrier. But we see this morning the blood-brain barrier in ALS is a little open, but still the brain biodistribution is low of phenofibrate.
Yeah. So those experiments were, first of all, this is preliminary results that I just started to test out on our model, and it's just been done on zebrafish models. So maybe in the future we can explore other options.